Our next speaker is Dr. Marwan Sabbagh, if I am saying this correctly. Please excuse me in that regard.
That's
Dr.
okay.
Marwan is a board-certified neurologist and a geriatric neurologist. There are very few in that respect. He's considered one of the leading experts in Alzheimer's disease dementia. He has dedicated his career to finding treatments for Alzheimer's and other age-related neurodegenerative diseases. He's the leading investigator for many prominent national Alzheimer's prevention and treatment trials. He's on the editorial board of the Journal of Alzheimer's Disease and Alzheimer's and Dementia. He's the chief editor of Neurology and Therapy. He has authored and co-authored almost 400 medical and scientific papers and articles. And I'm going to avoid telling about the number of books. He has written number of books. He has edited number of books. I mean, it's almost half a page. If I go into that, it will cut into his time. The only thing I would mention is that today he will be talking to us how to evolve therapeutics in AD and how to envision usage of biomarkers for detection and screening. Dr. Marwan Sabbagh.
Thank you. Thank you for including me, and it's an honor to be here. And hello to my friend Murali Dorswamy. I am a neurologist by trade and a clinical researcher. I'm going to walk you through as the host has asked me to talk about therapeutics and then talk about biomarker applications. As you know, that therapeutics is an evolving field, and if you look at the purple line, we have lived in the symptomatic phase with the cholinesterase inhibitors and memantine for over 2 decades now, with the idea that they improve symptoms, but they don't affect the trajectory. Whereas the ideal scenario is in the era of DMTs is to move from the green dotted line to the blue line, because the truth is the future of Alzheimer's therapeutics is a chemotherapy approach. It's not a one drug, but it's maybe a 5, 6, 7 drug cocktail that hits multiple targets. People say there's nothing going on in this Alzheimer's space. This is just the phase 3 programs that have failed since 2003. This slide here represents $40 billion of investment, and that just suggests that there's an active area of research, but an area that still has an unmet need. Every year, as you know, Jeff Cummings publishes this paper, what I call the Wheel of Fortune of the Alzheimer universe. It's important to understand that there's 75 drugs in clinical trials that are being developed. If you look at the top third, that is the monoclonal antibodies, and in the inner circle is the phase 3 programs. We'll talk a bit about the monoclonals that you're hearing about, but you know that there are a lot of drugs that are in development that are coming and going. Even since this is the 2020 slide, the 2022 slide will actually have some drugs on this and some drugs dropped off of it. So this is a rapidly changing field, but there are a lot of small drug targets in the bottom left, right, and then symptomatic treatments on the bottom left. So let's, you guys have spent a lot of time discussing amyloid biology, but the question has been is where do you intervene and why should you do that? And what we've learned over time is that if you look from APP, when it's cleaved to become monomeric A beta, it turns out that the monomeric A beta is essentially a nascent protein, and that really the reaction starts to occur in the oligomeric species. That's when you get the microglial activation and inflammation, et cetera. And when you go from oligomeric to fibrillar to protofibrillar to plaque. And the reason I say this is that so far the data suggests that targeting monomeric A beta largely has not worked, at least in the symptomatic phase. So we need to kind of coalesce around the idea that we target at the oligomeric species phase or later. And when you look at amyloid biology, again, 2 decades or more, people have said either you go for production-based approaches or clearance-based approaches. So if you look at the left-hand part of the slide, production-based approaches is the secretase inhibitors. The gamma and beta secretase inhibitors have largely failed, whereas the clearance-based approaches, that's the monoclonals, are still coming along. And so we're seeing a lot of things about this. So I want to talk about the fact that active immunization was the first gen approach. And when we talk about the monoclonals like aducanumab, which we'll talk about here, you need to understand that before there was aducanumab, the first gen was an active immunization called AN1792. Was published what got people excited is the Nature paper by Dave Morgan, suggesting that you could clear out plaque when you actively immunize the patient, transgenic mice. Worked great, cured Alzheimer's. If I sat at this conference 20 years ago, I'd say I think we've cured Alzheimer's disease. They went into a phase 2 clinical trial, which was a 360 patient enrolled study. Typically, it takes a year to enroll. They enrolled it in 6 weeks. They started in this October. By January, we were hearing of 2001. By January 2002, we were hearing about some problems. And by March of 2002, that study was stopped because of safety, because there were 17 cases of encephalitis and 5 deaths. And if you actually look at the Nickel paper published in Nature in 2004, it showed, in fact, that the drug worked, that AN1792, in fact, did clear out plaques. And so the concept of amyloid removal was established at that time. And the idea was maybe active immunization is not the best way to go, and maybe we should go for passive immunization. So then the monoclonal started to come. The first generation monoclonal antibody was a drug called bapineuzumab. And bapineuzumab was a drug that caused something which we'll talk about here called ARIA, or amyloid-related imaging abnormality, what we called at the time vasogenic edema. But the point is that at the phase 1 trial dose, we were 5 and 10 milligrams per kilogram. But by the time we got to the phase 3 dose, we were using 0.5 and 1 milligram per kilogram. We dropped the dose 10x. And so what you see in this upper left-hand panel is flipped backwards. Sorry about that. But the bottom left hand you see is that amyloid was clearly removed as early in the first gen monoclonal antibodies of bapineuzumab. But the clinical efficacy signal was lost when you drove it down. So that suggested that we could hit a biomarker, but we couldn't hit a clinical therapeutic target. When you looked at the other competing drug, which was solanezumab, which targeted, so bapineuzumab targeted the plaque form of amyloid. Solanezumab targeted the monomeric form of amyloid. And so the idea, so this is upstream to bapineuzumab, and the concept was that did not move amyloid at all on an amyloid PET, but had a small, very, very small clinical effect. And the p-value was just not sufficient for the drug to move into approvability, despite 3 trials, Expedition 1, 2, and 3. And Lilly has stepped away from solanezumab, except that they're still investing in their A4 trial, which is an asymptomatic treatment. So the concept, of course, is that bapineuzumab cleared amyloid, did not have a clinical efficacy signal. Solanezumab did not clear amyloid, but had a small efficacy signal. So we did not have convergence on biomarker signal and efficacy signal. Enter into aducanumab. And aducanumab is a different monoclonal antibody insofar as it's a natural antibody derived from humans that people have natural immunity to amyloid. And so they derived the natural immunity to amyloid and created this monoclonal antibody. The reason people got excited was this study here, published in Nature. Savigny, you know, and I know publishing anything in Nature is next to impossible, and publishing a clinical paper in Nature is next to nearly impossible. You could win the lottery more likely than you could publish a paper in Nature, a clinical paper in Nature. But this is the paper that really set the world on fire. And why is that? If you go from left to right, you see in a dose-dependent manner that the higher the dose, placebo 1 milligram per kilogram, 3 milligram per kilogram, 6 milligram per kilogram, and 10, in a dose-dependent manner, removed amyloid. And you see that graphically on the right. You see that the placebo group is the top image, and the 10 milligram per kilogram is the bottom image. Red represents amyloid, and you see very clearly that amyloid is removed, gone. It's out of the brain. And this is the first time you also see in the dose-dependent manner that the higher the dose, the more stabilization on the clinical decline, CDR, clinical dementia rating scale, mini-mental. So this was the study that showed that there was a directional concordance, meaning that in a dose-dependent manner, the higher the dose, the more amyloid was removed and the more slowing of the rate of decline. Now, somebody's going to ask me, amyloid and clinical decline do not correlate well, and that is correct. But maybe it might be a secondary mechanism of action which would be relevant here. Fast forward to phase 3 trial, read out in 2019, published in 2021. This is the drug that got everybody excited and upset at the same time. And this is important to understand. In the left-hand side is the EMERGE study, which showed that in the purple line, you see that they slowed the rate of decline very clearly, about 30% slower than the gray line, which is the placebo. I guess that's a green line, is the placebo group. The ENGAGE study on the right-hand panel showed a similar trend, but did not meet its pre-specified statistical endpoint. And yet the FDA filing was around the removal of amyloid, and they got the approval not on the clinical efficacy signal, they got the approval on the removal of the biological target. And so that's where the controversy is the pundits who opine don't and say that the drug doesn't work are opining on the fact that the drug did not hit on one of the 2 trials. But the clear efficacy signal is still being established. And I have patients myself treated with aducanumab 5 years out are doing pretty well, stable in their slowing the rate of decline. So this is what is the drug that has been approved. Now, the thing that is important, when you can weigh in all you like on this, you need to understand that this is the first of 4 drugs. This is second generation drugs that are now targeting monoclonal antibodies targeting amyloid. The second one is a drug known as BAN401. Lecanemab is its new drug name. Lecanemab is also a monoclonal antibody, 10 milligram per kilogram twice monthly, every 2 weeks, showing that it can remove amyloid 90%, and that when you do that at 10 milligram per kilogram biweekly, you reduce the rate of decline by up to almost 50%. And this is now in their phase 3 study called the Clarity study. This should read out later on this year. Third drug in the class is a drug called donanemab, a monoclonal antibody also. But this one, instead of looking at oligomeric or protofibrillar species like the other 3 in the class, hits the pyroglutamate epitope of the plaque. And so the reason this is important is the pyroglutamate epitope of the plaque seems to be something that correlates very robustly. And what you see in the right-hand panel in the green line compared to the gray line is it slowed the rate of decline a lot, more than 30%. So this is important because this is the composite study called score called the IADRS. This was published in the Minton paper in New England Journal last year. But I want to say a couple other things about this drug that people got very interested. One is that they selected not only the presence of amyloid, but they selected on the level of tau. And tau, the patients had to have intermediate amounts of tau. Too much tau was considered not movable, and low tau was considered not sufficient for progression. So they picked intermediate amounts of tau. Second thing is they did what will probably become what we call induction therapy, where it caused patients that once their amyloid was removed completely, they stopped treatment. And then they followed these patients. And I think this is the first gen induction therapy approach and could be very useful in the future. The fourth drug is a drug called gantenerumab. This is the Roche drug. And you see, again, very, very clearly removes amyloid, very, very robustly removes amyloid. Their phase 2 trial called Marguerite Road AD is used 225 milligrams dosing. They feel like they missed on their efficacy target, and they have now done their phase 3 graduate study, which has increased the dose to 1,200 milligrams. They will read out in the next 6 to 9 months. The advantage of Roche gantenerumab over the other 3 in the class is this is a subcutaneous formulation, which could be very appealing to a lot of patients. What have we learned? Is that the efficacy signal is going to only be derived in the patients in the very mildest phase. That means MCI and mild AD dementia. You're going to do risk stratification on the basis of the APOE4 genotype. You're going to, we need to understand that there's a major risk factor or complication called ARIA, amyloid-related imaging abnormality. There are two types of ARIA. One is ARIA-E, which is vasogenic edema, and ARIA-H, which is microhemorrhage. Most of the time they resolve. I'm going to show you the examples of ARIA-E and ARIA-H. These are representative. They're right off of Google. These are not actual patients that I've seen. And so I would tell you that don't pick, this is a patient who would not get a monoclonal antibody because this patient has a lot of microhemorrhages. The rule of thumb is if they have more than 5, they should not be getting a monoclonal antibody. Again, this is just taken for purposes of teaching. Vasogenic edema, this is again taken off the internet. You can see vasogenic edema and shows most of the time these are clinically asymptomatic, 75% of the time clinically asymptomatic. And when it is symptomatic, it doesn't require anything but stopping the dose, watching the patient. You can even dose afterwards, maybe at a lower dose. So safety MRIs are going to be required. The other people talk about is tau and tangles, and that makes a lot of sense. And there have been multiple approaches, including kinase inhibitors, microtubule stabilizers, assembly inhibitors, and immunotherapy. So the idea for the immunotherapy is that when the phosphorylated tau, when the neuron dies, these little forms of phosphorylated tau are released and taken up by adjacent neurons in a process called preonosis. And the monoclonal could bind those free forms of phosphorylated tau to prevent the spread. It works in theory, but it hasn't really worked very well in practice. So 3 monoclonals have stopped moving past phase 2. And so there's a lot to say about the tau space. I think this is stay tuned. Again, a theoretically appealing application has not moved forward. I wanted to tell you there's a lot of things that are going on beyond that, including sleep, a drug called Sorexavant, which is a drug now approved to treat sleep in Alzheimer's disease. We're seeing this approach where we're starting to carve out, instead of going for all the disease, we're trying to do something called segmentation, and the ALZ 801 is selecting patients on APOE4 homozygotes. These are patients who in their phase 2 trial hit the pre-specified endpoint. Then there is the hepatic growth factor ATH1017, which synaptic trophic factors and NMDA and antagonism, and then the gengibane inhibitor atuzanginstat, which just read out their phase 2 trial with a little bit of a safety issue, but showed that they could get some slowing the rate of decline in moderate Alzheimer's disease at 40 milligrams twice a day. The second ask was about biomarkers, and that's a good segue into where we want to go, because the questions are going to be, how do we pick patients in future, both in clinical practice and trials? You know, historically, the Alzheimer's approach has been a diagnosis of exclusion. So the idea is that you heard in medical school and you heard in graduate school, you could only diagnose Alzheimer's disease with an autopsy. And that was based on the NINDS ADRDA criteria of 1984. But it was recast in 2011 as NIAAA working group, which said that biomarker evidence was considered to be suggested but not essential, but that, and it recapitulated the 1984 criteria. What happened then is classification of Alzheimer's disease moved and split. So IWG is the International Working Group. This is Europeans and Canadians, suggesting that if you have progressive amnesic disorder plus any biomarker, APOE, CSF, PET, anything, you have Alzheimer's disease. Whereas NIAAA Working Group stuck to the clinical phenotype and said that biomarker evidence was considered to be advisory but not essential. So the reason this has been important is up until last year, we would say we would do a clinical phenotype of the patients, but not biomarker confirmation beyond excluding other pathologies. And so that's changing now in the era of aducanumab. So the reason this is also important is that the ATN criteria has been put forth by Cliff, Jack, and colleagues. A stands for amyloid, T stands for tau, and N stands for neurodegeneration. And the key you need to understand is you cannot have Alzheimer's disease. I'm very careful in my use of words, disease without amyloid. You must have amyloid, but you can have levels of tau and neurodegeneration. If you don't have amyloid, it doesn't mean you don't have a neurodegenerative process. You can have tau positive, neurodegenerative positive. That just means you don't have Alzheimer's disease. You may have a tauopathy, PSP, CBGD, something else, but it may not be Alzheimer's disease. And so you say, why do we need biomarkers? We don't need biomarkers. Clinical diagnosis is sufficient. Well, not really. I led a study on florbetaine. This is the Neuraseq amyloid imaging compound. We followed 57 people with clinical diagnosis to autopsy, and turns out we experts, world experts, were wrong 23% of the time. The clinical accuracy was wrong, was right 77% of the time, and wrong 23% of the time. And if you actually look at this down below, the super experts, it's 66% of the time. So imagine a clinical diagnosis that is wrong 1 out of 3 times. That's where we're at in the field. That's why we need biomarker confirmation. So how do we get to biomarker confirmation? Well, historically, it's been a diagnosis of exclusion, as you see on the slide, but now we need to move it to a diagnosis of inclusion. And so the mandate is to come up with a biomarker specific, and you got to look at biomarkers in 3 general categories. One is they can be binary, positive, negative, which would be a diagnostic. 2 is a reflection of disease progression, and 3 is a theragnostic biomarker, which would reflect a disease target engagement. So there is now PET imaging, which is approved, not reimbursed, CSF testing, which is approved and reimbursed now. Just in the last year, you're seeing it routinely used. And so we have all these biomarkers that are available to us. People ask me, why don't you just use MRI? Well, the answer is the specificity and sensitivity is very poor. If you look at volumetric, neuroquant is excellent for quantifying hippocampal volume below the 5th percentile as a proxy marker of neurodegeneration. But using MRI to differentiate one dementia status from another is not very accurate. I myself am a big fan of APOE genotyping. Historically, people have been very reticent to do so and have not. But why is that? Because amyloid PET is $5,000 US, and APOE genotyping $99. We know that if you are an APOE4 patient carrier and you have symptomatic MCI or dementia, the probability you have amyloid in your brain is around 96%. So it's a very cheap, quick way as a proxy marker of amyloid, and it is age dependent. The last thing I want to say is that biomarker confirmation, when you look at those criteria, if you look at the left IWG, that means if you have amyloid in your, if you are biomarker positive, the probability of clinical progression using Kaplan-Meier survival curves is overwhelmingly high compared to patients who are biomarker not confirmed. Amyloid PET and tau PET are both approved. They're not reimbursed. Amyloid PET, $5,000 US, tau PET, $9,000. Amyloid PET is a binary, positive, negative, does not correlate with clinical progression, but is a marker, very robust marker of amyloid. Tau PET correlates very well with clinical progression. Approved is also called fluortaucipir, also known as Tauvid, but not used in clinical practice routinely because of its expense. And here's the good example that when you look at Braak staging, tau correlates very well with clinical progression, and Braak staging does as well. And tau PET recapitulates Braak staging exactly in vivo. So Braak was right. So the imaging has been, the competition in the last several years is discussion on whether you should use imaging biomarkers or CSF biomarkers. And the answer is that we routinely in clinical practice are using CSF biomarkers more than we are in amyloid biomarker, than imaging biomarkers. Looks like I'm past my time because I see the speaker at the podium. So I'll stop there.
Yes. Please go ahead.
Okay. Okay. So I'm saying CSF biomarkers are very accurate. The specificity and sensitivity of CSF biomarkers are overwhelmingly fantastic, and they measure tau and amyloid and correlate very well. There's been multiple studies. We know that it correlates very well with amyloid PET, and it is a good marker. If you're MCI, the probability of progression to dementia is very high. And I just said this. There's a new plasma amyloid test called the Precivity test, which is an A beta 42 to 40 with an APOE genotyping. This uses the mass spec platform. I will say to you that the Precivity test is one of several that could be coming on board. One includes the Samoa platform and the MagQ IMR Taiwanese platform. So I think this is the first thing. The question that has been discussed is, will it be sufficient as a diagnostic, or will it be a screening tool? Is it the negative predictive value that excludes pathology, but you have to do tier 2 diagnostic confirmation with amyloid PET or CSF testing, or will it be sufficient enough to use as a treatment indicator? These are to be determined. A lot of people think that plasma amyloid will not be the final say. It'll probably be p-tau 181 or 217 or 231, or a multiplex assay, which hits multiple biological biomarkers. And with that, I wanted to talk about a new algorithmic approach. This is the way we're going to go. The biomarker confirmation will now become essential to clinical practice, not an aside issue. So I just want to bring this all to your attention. And I want to say it's a rapidly changing. We understand that Alzheimer's is going to go from a terminal disease to a chronic disease in our lifetime. This is the diabetes and HIV of our lifetime. There are 75 drugs in development, and we're in a transformative moment in the use of biomarkers. Thank you.
Thank you very much, Mark. It was excellent talk. It really trying to cover it in half an hour was amazing.
There is an online question.
We may have a couple of minutes, or at least and let me begin by just asking you a question. It all sounds so interesting, and you talked about this how all these new autoimmune and antibodies and treatments, how would it affect in terms of economically? We already know, we've heard about it, how Medicare will be affected. And so it can have a major impact overall. So where do you see where and when all these interesting interventions may become routine?
Yeah, that's a good question. And that's a question that a lot of people are asking. And this is a complicated question. What we do know is, despite this worry that we're going to bankrupt Medicare, you're only talking about 20% of patients walking in the door will ever get a monoclonal, will be eligible for a monoclonal antibody. So it's actually going to be a niche drug. It's not going to be everybody getting it. This is not the drug that bankrupts Medicare. You know, if you're talking about 6 million people, 20% is 1.5 million, of which then there'll be even less than that. So the majority of patients will actually not get these kinds of treatments. Second of all, for a variety of reasons. Second of all, we probably see a variety of different targets. And so induction therapy might be in the future that we give this, like we would a chemo drug, you load it up, clear their disease out, and then put them on some kind of low dose maintenance. What I'm saying to you is that we can't say this drug will be the disaster, economic disaster that has been posited.
We may have time for one more question from the audience, if there's one. Is there any question in the chat, if you might? No. Okay. So in the interest of time, I would very much like to move on.